Hi, everyone. My name is Isabel Torres.
And my name is Sharn Basi, and we are students from CSU East Bay doing a project on methane uptake in compost-amended grasslands in the Concord area. Our project is an investigation that combines trace gas measurements, quantitative PCR, and soil microbial analysis to show the impacts of compost application on rangelands. The goal is to analyze the response of microbial communities that consume methane. In order to meet our carbon net neutrality goals and sustain growing populations, we need massive removal of greenhouse gas emissions from the atmosphere. And one of the ways to do that is to use nature-based climate solutions, NCS. Now, NCS approaches are predicted to be able to remove 125 million metric tons of carbon, and this can help states like California achieve its 2045 carbon net neutrality goals. Putting carbon back into the soil not only boosts soil productivity, but it increases carbon accumulation over time, which reduces our carbon footprint. Compost was applied in October 2020, and for 1 year preceding and 1 year following the compost application, we used multi-scale measurements at the site to quantify carbon and methane fluxes. We measured those greenhouse gases using eddy covariance methods, as well as a permanent soil flux chamber. These two methods measured carbon exchange within the ecosystem, and then we also used a manual chamber to measure during site visits. The manual chamber measured carbon and methane at the same time. We also processed soil for organic carbon and nitrogen using an elemental analyzer. In October of 2021, we took and froze soil samples to be analyzed using quantitative polymerase chain reaction, qPCR, to see how the microbial community was changing in response to the compost. Specifically, we're testing for the abundance of the PMOA gene, which would be abundant in samples with a lot of methanotrophs. Compost amendments increase uptake of carbon and are shown to enhance soil fertility, soil moisture, and stimulate plant productivity, as well as increase carbon deposits in the soil. And these increases are hypothesized to extend the growing season, which allow for greater amounts of carbon to be sequestered. We hypothesize that a higher soil moisture, carbon, and nitrogen content caused by the compost application will lead to soil conditions that are favored by methanotrophs, which are the microbes that consume methane in soils.
What were the results? The compost application led to carbon inputs available for storage and increased soil microbial activity. Studies have shown that carbon inputs may alter soil microbial diversity and can be more important for controlling carbon decomposition. These microorganisms play various key roles in carbon sequestration, such as soil organic carbon stocks through the formation of microbial biomass and the stabilization of its residues associated with specific minerals in the soil. This is known as the positive priming effect. Figure 1 on the left shows carbon 2021 treatment versus control soils. We have treatment type on the x-axis and organic carbon percentage on the y-axis. We can note that the treatment soils are significantly greater in carbon than the control. Okay, figure 2 on the left is giving a time series of GCC from phenocam footage in treatment and control plots. The x-axis is our day of year, and y is measuring GCC, which is the green chromatic coordinate of a photograph. These photographs were processed in MATLAB, and as you can see, the treatment green-up began around day 375, around about 30 days earlier than the control green-up, which was after day of year 400. Figure 3 on the right shows treatment and control plots in the early green-up. On the right, we have our treatment green-up, and on the left, we have our control plots that aren't very green. Okay. The compost application also led to increased uptake of methane and increased fluxes of carbon dioxide. We hypothesized that soil nutrients in treatment soils created favorable conditions for the methanotrophs, and these are the soil microbes that uptake methane from the atmosphere. So here we have 4 graphs available for us. On our x-axis, we have our day of year, and our y-axis, we have soil moisture, soil temperature, average methane flux, and average carbon dioxide flux. This brown arrow indicates when the compost was applied, and soon after you can see all of these negative fluxes, which indicate an uptake of methane occurring, and we also see a higher increase in carbon dioxide being emitted into the atmosphere following the compost application. We also plan on taking qPCR measurements of soil samples that were collected in September 2021. In conclusion, the compost applications to rangelands have significant impact on phenology of the ecosystem. Some of these scale emergent processes that weren't accounted for in previous model simulations can now be further investigated. The earlier green-up and longer growing seasons have implications for more water cycling and the need for cattle farmers and land managers. Soil organic carbon measurements do show a significant increase in soil organic matter in surface soils, and methane uptake was significantly increased after compost application. Okay, why is it important to continue this research? Oftentimes, the role soil microbial communities, also known as SMCs, play in soil carbon sequestration, also known as SCS, is overlooked. So this study helps characterize soil microbial communities of grazed grasslands and compost managed lands that can be applied to modify farming practices to mitigate climate change. Also, this study highlights the importance of soil health in agricultural and land management processes. And in order to improve the sustainability of soil management, it is crucial that the impacts of agricultural practices on soil microbial communities and soil carbon sequestration are quantified and understood. All in all, if compost application has this much of a positive effect on microbial activity, then future large-scale applications can potentially enhance methane uptake in working lands. These methods are exactly what we need to be exploring in order to achieve our goals toward zero emissions.
We want to recognize all the professors that were involved in our project, Dr. Patty Oikawa, Dr. Chris Baysdorfer, and Dr. Hausen Chu, as well as grad students and lab members who worked with us in the lab. Thomas Fenster, Jacqueline Marroquin, Tiffany Hopkins, Maya Matsumura, Julie Shahan, Ola Oluwatobi Thomas, Serenity Pena, and Leilani Rains. Before we end our presentation, we'd like to open up the forum to questions.